Hello my dear students, welcome to the MS Science Academy. Myself Meenu Srivastav, and today we are going to discuss about the clean area classification. Okay, so the clean area basically a room in which particles and the viable count is kept with the standard and defined range. Okay, so in the clean area the particles maintained up to a certain limit. Okay, so here we can see there are so many standards by which we can determine our particle size. Means up to this area we can maintain the particle or the particulate material up to this range. Okay, so according to this we will see there are so many types of the clean area classification according to the grading system, according to the 100, 1000 and so many systems. Okay, so let's have a look. Now it's important for manufacturing of sterile and pharmaceutical product. Why is it necessary to maintain the clean area or the aseptic area and aseptic area? Because we have there are so many sterile preparation, pharmaceutical preparations, in the semiconductor preparations, so many electronics preparation in which we have to maintain the particle size up to a certain limit. So for that we have to maintain the clean area or we have to just maintain the clean area. Okay. Now there is a federal standard 209E section. The federal system, that is the federal system, according to the federal standard, they gave the one standard definition, that is they define the clean room. They define a clean room as a room in which the concentration of airborne particles are controlled to specific limits. Okay, so the concentration of airborne particles are controlled to a specified limit. Okay, now second is the British standard. British standards also defined a clean room. They gave a specific definition. Okay, British standard defines a clean room as a room with control of particulate contamination, constructed and used in such a way as to minimize the introduction, generation and retention of particles inside the room, and in which the temperature, humidity, flow pattern, air motion and pressures are controlled. It means that there are so many factors by which we can maintain the clean area. Okay, there are so many factors like temperature, like humidity, like air flow pattern, like air motion, pressure also there. Okay, so there are so many factors by which we can maintain, we can introduce, we can retain the particles in that particular area, so we can do our preparations. Okay, how we can maintain the clean area? Okay, the clean area we can maintain by the UV ultraviolet radiation. Okay, by which ultraviolet radiation. Second, as we have discussed in the earlier previous lecture, that all the clean areas or the aseptic areas having the airlock entry portals. Okay, there is a double door entry system by which we can maintain the airlock system. Okay, special filtrations, or we can say when the air will be introduced into the aseptic area, there are the pre-filters or filter pad assemblies are associated with the HEPA filter or that aseptic room area. So for that, the air for the upcoming air, the air will be filtered very accurately manner. Okay, now the sticky mat for removing the particulate matter from the shoes. Okay, if any room before each and every room there is a sticky mat are present, so by which we can remove our dirts or that other particles which are attached or just stick with our shoes and other footwears also. Okay, now the fifth point, that is the positive room pressure. What is the positive room pressure? Suppose that in that clean area the pressure is high and that beside the other room the pressure is the low, so automatically the air moves towards the high to low pressure. Okay, so automatically air moves from the clean area towards the other area, so there is no chances for germination of germs with the help of air. So here the clean air classification, so in the clean area room or clean area premises, the air pressure should be high in that particular area as compared to the besides area or beside sites. Okay, So here the automatically, if the pressure is high in the clean room, then the air will move towards and the air will move from the clean area, and that the particulate material will be reduced automatically. Okay. Now the clean area classified as per the WHO guidelines. There are so many guidelines here. One guidelines we will study according to the WHO guidelines. Okay. According to this, the clean area classified grading A, grading B, grading C and grading D. Okay. So for that, the maximum permitted particles per meter cube. Here the area is a per meter, volume is a per meter cube, equal and above, means the particle, suppose that 3520, so the particle range it's equal to the 3520 or maybe some above 3529, 3528, like fluctuation will be possible. Okay. Now the second term is that at rest and at operation. What is at rest? It means if we are not doing anything or any just activity are not doing in that particular area, that time it's a rest condition. In operation, it means that the part that there will be any activities are going on, like packaging, like filling and other activities are going on. So that time it's known as the in operation. Okay, means there are some some activities are going on. Here now in the grading A, here at rest 0.5 micron meter and 5 micron meter. There is a range 0.5 to 5 micron meter. There is range 0.5 to 5 micron meter in operation and at rest. Okay, in grading A, 0.5 micron meter is 3520. Okay, 0.5 micron meter particle size per meter cube equal 3520 or maybe above. Okay, and 5 micron meter 29 at rest for grading A. Here in the operation, same same particle range is there 3520, and for 5 micron meters 29. Now grading B, it's 35200. Okay, 0.5 micron meter 35200 and 5 micrometer 293 particles. Okay, in operation 0.5 35200 and for 5 micron meter it's 2930 particle. Okay, here the grading C 352000. Okay, for 0.5 micron meter and 5 micron meter is 2930. Now it's 0.5 micron meter 352000 and 5 micron meter is 29300. Here for the grading D, here the 0.5 micrometer is that 35,20,000 particles per meter cube equal and above. Now 5 micrometer it's 29,300. Okay, 29,300 equal or maybe above. Okay, now for the in operational condition 0.5 micrometer and for 5 micrometer it's not defined. Okay, till it's not defined, maybe it will define. So if you, here when I am preparing notes, then they are just suggesting it's not defined. That's why I have written here. Now another classification is there, that particle size 0.5 micron, less than 0.5 micrometer per feet cube, and the microbiological limit colony forming unit, that is a CFU, colony forming unit per feet cube. Okay, so here the clean area classification according grading 100, 1000, 10,000, 1 lakh, like so. Okay, so here the clean area classification is grading is 100. For that 100, the particle size for less than 0.5 micrometer per feet cube is that 100, and the colony forming unit per feet cube is less than 1. Okay, here the clean area classification for 1000, suppose it's a A and here is the B. For 1000, the particle size less than 0.5 micrometer per feet cube is 1000, and microbiological limit colony forming unit per feet cube it's less than 2. Now the C, that is the 10,000. Here the particle size 0.5 less than 0.5 micron per feet cube, that is the 10,000, and microbiological CFU per feet cube, that is the less than 3. Here the third D classification, it's a 1 lakh. Okay, so 1 lakh, so particle size is less than 0.5 micron per feet cube is the 1 lakh, and the microbiological limit colony forming unit per feet cube is less than 25. Okay, so here there are so many classification. We will also discuss about the ESO system, that ISO system, and other classification also we will see. Okay, now the operation of the clean and aseptic rooms are recognized by 3 main areas. In the clean and aseptic areas, there are the three main zones. You can say three main rooms, three main compartments, or three main zones are there. For that, we will divide according to their according to their microbiological phenomena, or we can say microbiological cleanliness. Not microbiological, according to the cleanliness we can divide. First is a black or dirty area. As the name suggests, here in that area the particle and microbial control is almost impossible. Particles and microbial control is almost impossible, because there are so many interaction will be there, and there are so many persons are coming and going. So such areas, in that particular such areas, we cannot maintain the microbial control. Now the second is the gray and semi clean areas. The high standard hygiene and cleanliness can be maintained. Okay. Now the third areas, the white areas, the full hygiene, full control system can be maintained. Okay. So here the black and dirty areas, no control, and the gray, or we can say it's a semi clean, so controlled up to the certain extent. But in the white areas, the environmental control can be maintained for full hygiene control, or full environmental are maintained up to at a very high level. Okay. Now we will see what are the other grading systems or other classification. Now come to the classification of room. There are so many standards by which we can classify our clean room. Okay, so here one is the ISO 146441 standard. In that system, the particles count per cubic meter. Okay, per cubic meter, or we can say meter cube also. Okay. Now the second standard, that is I will denote it's a one standard, and second is the FS 209 system. In this system, the particles are count per cubic feet. But nowadays, the FS 209 system is not in the trend. It is replaced by ISO system. Okay. Third system, that is the EU standard. Okay. That is the EU standard, and the EU standard also divides the particle side, divides the clean room according to the grading A, B, C, D, and like so. Okay. According to the ISO, C stands for clean. Clean rooms are classified into 9 classes. Okay. According to the ISO system, the clean room classified according to the 9 classes: class 1, class 2, class 3, class 4, class 5, class 6, class 7, class 8, class 9. Okay. In this system, in the ISO class 1, its grading as a cleanest grading. Okay. It's a cleanest room. Okay. It's a cleanest area, and that ISO class 9, it's the dirtiest area. Okay. For that ISO class 1, it's the cleanest, and ISO class 9, it's the dirtiest area. Okay. In the pharma industries, according to the ISO system, we maintain the just particle size or the microbial unit in according to the 5, 6, 7, 8 class. ISO class 5, ISO class 6, ISO class 7, and ISO class 8. Okay. So in the pharma industries, we maintain the clean room extent up to the ISO 5th to 8th classification. Okay. Now the EU system, according to the EU system, the grading, the clean room is classified into the grading A, grading B, grading C, and grading D. Okay. According to FS 209 system, the classes are divided into the class 100, class 1000, class 10,000, and class 1 lakh also. Okay. Now here the classroom, the clean area, according to it's here AC, it's according to microbial count and air changes. Okay. The clean area according to microbial count and the air changes are classified as here. According to this, the clean room is classified as grading A, B, C, D, and here what are the criteria we have taken? That is the air sample. In that air sample, the colony forming units per meter cube. The colony forming unit means the how many microbial flora is present in that particular area per meter cube. So that this much colonies of bacteria or the microbial system will be produced. Okay. Now second is the settle plate. For that, Petri plate's diameter is 90 mm. The diameter is the 90 mm. 90 millimeter diameter Petri plates are used, and the colony forming units per hour we will observe. Colony forming units for the 90 mm Petri plates per hour we will observe. Next is the air changes per hour. Air changes per hour we will see, okay, for that another criteria. Now according to in the grading A, the air sample, in the air sample the colony forming unit per meter cube is the less than 1. The settle plate, here the colony forming unit per hour, per 4 hour is less than 1, and the air changes means the how the air rotation changes per hour, it's 240 to 480 waves of 240 to 480 times it will changed. Okay. Now the grading system B, the air sample colony forming unit per meter cube, it's the 10. Okay. Now the settle plate, it's the colony forming unit per 4 hour, it's the 5, and the air changes rate per hour, it's 150 to 240. Now the grading C, the air sample colony forming unit per meter cube, it's 100, settle plate, it's 50, and air changes per hour is 60 to 90. Grading D, air sample colony forming unit per meter cube is 200, settle plate, for that settle plate, the CFU per 4 hour is 100, and the air changes per hour is 5 to 48, means the air rotation is changes 5 to 48 times. Okay. Now the other classification, in that other classification, the maximum number of particles per meter cube we have taken in the range of 0.5 to 5 micrometer or less than micron, more than micrometer size. Okay. Now the second criteria, maximum number of viable microorganism per meter cube. Okay. So here I will just write it down like this. This is the per meter cube. Okay. So here for the grading A, it's 3500 for 0.5 to 5 micrometer, means this much particles per meter cube are permitted, and more than micrometer size, it's none. Okay. And the maximum number of viable microorganism per meter cube is less than 1. Grading B, in grading B, the particle size, the maximum number of particle per meter cube is the 3500, here the none, and the maximum number of viable microorganism is 5. In the grading C, here the 350000, 3 lakhs, okay, 3,50,000 for 0.5 to 5 micrometer, and 2000 more than 2000 as compared to micrometer. Okay. Maximum number of viable particles is here 100, and for grading D is 35 lakh, 0.5 to 5 micrometer per meter cube in the per meter cube particles, and 20,000 particles more than meter cube, more than micrometer size, and the maximum number of viable microorganism, it's 500. Now, how we can monitor our clean room? There are certain criteria by which we can monitor our clean room at a regular interval, so that we can maintain all the other grading system and all other the criteria, so that we can just maintain the cleanliness and other just maintain the other criteria also. Here the clean room maintenance, for that particles monitoring in air. Okay, for that frequency, here I have written the frequency also. Okay, here is the frequency also. For the particles monitored in air, for the particle present in the air, we can monitor for the 6 months. Okay, for the 6 months. HEPA filter integrity means the HEPA filter testing yearly we have to do it, means the continuously HEPA filters we have to use in the microbiology lab or the clean area, so we have to yearly check the HEPA filter. Now the air changes rate calculation means how much air flow, air pressure can be maintained, not air pressure, it's air flow. Okay, so air changes rate calculation for the 6 months. Okay, temperature and humidity we have to calculate daily. Air pressure differential means the positive pressure, negative pressure in the outer side, that we have to just measure the daily. Now the microbiological monitoring by settle plates. Microbiological system means how much microorganisms are present in the particular area is also maintained or also observed daily. Okay, so this is the complete clean area classification. I hope you have understood my lecture. Any doubt you can ask me in the comment section, and please like, share and subscribe, and soon we will start the BSc Biotech syllabus also, so don't worry about the BSc Biotech student, we will soon start that syllabus also. Thank you.